Hey guys, we're going to do a video today on active transport, which is the last topic for this chapter. We just went through examples of passive transport, so I've been kind of reiterating the fact that passive transport, as soon as you think passive, you've got to think no energy requirement. Because it's no energy requirement, it can go from high to low, and it's a spontaneous idea where we talked about diffusion, osmosis, facilitated diffusion, and for some classes we talked about diffusion through ion channels. And so today we're going to flip gears and we're going to go to active transport. And so the idea of being active in something is a requirement of energy. So now you have to associate anything that we're talking about today is going to be specifically needing energy to accomplish this process. And also, when you think of passive transport, we've been going from high to low concentration. Other words I talked about, you can go down the concentration gradient. With the concentration gradient, they all mean the same exact thing. In active transport, you are going to go against the concentration gradient, saying you're going from a low to a high. You're going against that grain that you would actually be thinking would require no energy. Now we need energy to go opposite that, going against traffic, from low to high, or they say against the concentration gradient or up the concentration gradient. Those all mean the same exact thing. So there are a few different examples of active transport we will talk about. Behind me is one example. What I think is going to be easiest for you to get a background idea, I'm going to ask you to pause this video and go into Google and search sodium-potassium pump. Sodium and potassium, two different words, and pump, P-U-M-P. If you want to add the word animation after it, it'll be the first link. And the way that you know that it's the proper link is in the web address, it'll say like McGraw, I think it's highered.mcgraw. If you see that, you should recognize it. It's the same animator that I've been showing in class. What I want you to do is right now watch the animation of the sodium-potassium pump. So you get an idea. Press pause. Okay. All right, hopefully you watch that video and it makes a little bit of sense because now I'm going to explain that video step by step. So behind me, I'm going to turn the lights out because I know with the lights on, it's a little hard to see the detail. So I'm going to flip the lights out. This will come into view a little bit more and we're going to go through the process. So this is active transport because what's going to happen is the molecules we're looking at, which are sodium and potassium, will be going against their concentration gradient. So let me turn the lights. So in your notes, you have a membrane, and in it I have kind of removed 4 different areas. So I'm going to pick 4 of these, and I'm going to say, try and mimic the diagram. The first one is a pretty important one. So what you see is the label. This is inside the cell, so make below your membrane labeled as the inside of the cell, which would be the cytoplasm. Above the membrane, that is extracellular or outside the cell, one and the same thing in their meaning. Notice the protein pump. This is a pump. Usually when you associate or see or hear the word pump in action of a protein channel, a pump means you're using energy. So this protein is going to require energy. At this point, we see the interior of the protein channel has 3 locations for sodium, which is the chemical symbol Na, for it to bond. On the right-hand side, we see 2 locations with our triangle based, which are going to showcase how the potassium. So this protein channel allows both sodium and potassium to go in and out. So we're going to look at the motion right now. At this point, the sodium and potassium pump is open to the inside of the cell. So when you're going to draw it, it's going to kind of draw it in this fashion so that the inside of the triangle, if you can look here, here's a triangle, the open end is exposed to the inside of the cell. So draw that on the first area on your phospholipid bilayer, furthest to the left. Draw 3 sodiums that are bonded to the inside of the protein channel. Notice right now, the energy molecule, ATP, is right here. There's the A, and then there's 3 Ps, which is the triphosphate portion. It is not connected to the protein channel yet, which means when there is no energy supplied, the protein is shaped as such. If you actually had to do this, this is very simple. It probably doesn't require any energy. You can do this for days. So what we're now going to do is progress to, once the 3 sodium have connected, we now are going to connect the ATP to the protein channel. This gives it the energy so it can transform the conformation from this shape. So now it's going to connect here. We're actually not going to, I don't think this is a necessary diagram to draw. So we're going to skip over the letter B and I want you in the next spot over in the membrane to draw this letter C diagram. Here's the phosphate, which supplies that energy for the protein. What now it does is it transforms the connection from here to now going like this. Imagine that this was, once again, the formation. This requires no energy. If you had to connect to your elbows and connect them and kind of do a V with it, it actually does require a little bit more energy to do. I can't connect, and it probably, for some of you, depending on how much strength you have, it's going to be a little bit more difficult to do that and hold it for a long period of time. Maybe you can do this, but to actually open it up to a V does require some energy. That's how I remember that kind of idea. This requires energy to fit in this conformation. So the phosphate group is attached, which allows the sodium-potassium pump to open. When it opens to the opposite side, which is outside the cell, the 3 sodium ions are going to now leave. At that moment, the potassium ions are going to seize the opportunity to bind to the open channel. So as it's still open to the other side, 2 potassium. Notice 3 connected for the sodium, but only 2 potassium actually can come in. So on the third diagram over, what you're going to do is you're going to draw this picture. So the phosphate is still attached, the connection is still open to the outside of the cell, and the potassium are going to come in and bind 2 at a time to this side of the protein channel. So once you have that, now the potassium are going to be transferred to the inside of the cell. Now how do we allow that to happen? This phosphate, if we remove it now, there's no more energy supplied. So the conformation of this channel, when there's no energy, it's going to go back to the original. So that's what happens here. In letter E, the phosphate is, you can see detaching, if you can see that detail. But notice what's happening to the channel. It's actually going to start to contract here, and this is going to close, which is going to open the other side. So here's the opening. The 2 potassium have binded. The phosphate is leaving. And as soon as it's completely gone, now it goes back to the original conformation. When it does that, the 2 potassium ions are going to now be put into the cell. So based on the movement, notice how this has to work. The sodium is going out. If this is active transport, that must insinuate that this is the lower concentration of sodium because it's going against the grain. It's going from low to high. So in all of our cells, the sodium ions have a lower concentration, this is important, inside the cell. Sodium outside the cell must be higher because it's going from low to high. The sodium's going in this direction. It's opposite for potassium. Since the potassium is going from outside the cell inward and it's still active transport, that must mean that there is a lower concentration outside the cell for potassium and a higher concentration of potassium inside the cell. I always abbreviate, and I kind of come up with my own mnemonic device, and that is going to be NaOKIN. Naokin stands for sodium going out, potassium goes in. Sodium out, potassium in. And that's how I keep on remembering the idea that it doesn't necessarily say that, it doesn't help knowing the 3, I gotta still remember that. Where 3 sodium go out and 2 potassium come in, but this starts you on that idea of, well, which direction do they move? Because this is constant. It's not like it changes. Every sodium-potassium pump will only pump sodium out and potassium in. Naokin. All right? So this is one example of the active transport. And I know that because it requires energy, and the movement of the ions is going to be against the concentration gradient. All right? So that is one example. The other example that we need to talk about is going to be 2 processes that are reverse of one another. They are called endocytosis, so flip the page, you're going to see this idea. Endocytosis. Endo means within. So what's going to happen is, this is a process by which molecules are going to enter into a cell via the cell membrane. So things that are outside, such as food, need to be taken into the cell. They can't squeeze in. They're obviously super large molecules because they're food. There's not even protein channels big enough for them. So what starts to happen is, this is the food right here. What will happen is the cell membrane is going to kind of collapse right around that food, and put it into a vacuole. So this vacuole now is the storage for the food. What will happen is this vacuole is then going to travel and combine with a lysosome. And why would it combine with a lysosome? Remember that lysosomes contain those digestive enzymes that break that food down. So this is a way for organisms to get food into their system without using the actual protein channels or diffusion right through. You don't see a direct connection to energy requirement because I don't see the word ATP here, but realize it doesn't fit the whole idea of low to high concentration. This is a process that fits under active transport just for the idea that the cell has to expend energy to do this process. So endocytosis is the bringing in of food. Once the food has been digested, the waste has to leave. So this is the second process of this active transport. It's the removal of waste, and that's done by exocytosis. The prefix exo meaning to release. So exo continues. This cell, the vacuole combination with the lysosome, now has a bunch of waste particles in it. We want to get rid of it. So what will now happen is this cell is going to fuse with the cell membrane, and the contents of the actual combination of vacuole and lysosome are then released out of the cell. Again, they could be possibly too large to get out via a protein channel. So what will happen is the membrane of this vacuole actually is the same membrane that now becomes a part of the cell membrane. It's a phospholipid. So if you have a lot of endocytosis occurring, you can imagine the surface area is going to become smaller for the cell because this cell membrane is now right here. So the surface area is collapsing and it's getting smaller. But then with exocytosis, it's being put back into the cell membrane. So constantly, your cell membrane is changing. It's always getting smaller, getting larger, things like that. It's not completely a static idea. All right? Two versions of endocytosis. Sometimes they say if this is food, they call it phagocytosis. It's just a specific type. I spelled that wrong. H-A-G-O, cytosis. Sometimes they say it's food and or large particles. And then if it's just liquid, they kind of call it pinocytosis. All right, those are just two variations, more specific types of endocytosis. There is not, it's like taking in food, taking in liquid. There's not specific terms for excreting liquid versus excreting food. It's not as detailed. It's exocytosis regardless of that. All right? So that's the information you need to know, and then we're going to kind of do some more review with it. But hopefully that made sense, and you can ask specific questions tomorrow in class. All right? We'll see you.